Okay, back to osmosis. Osmosis is water moving through a selectively permeable membrane to equalize the concentration of the non-water substance on either side. So water will move from a place where it's supposedly higher water to a place where it's lower. And this will dilute it. So water will move from a place where there's a lower concentration of non-water particles to a place where there's higher concentration of non-water particles, okay? So when you're paying attention to osmosis, it's about the non-water particles that cause the movement. So quite often you hear about pump salt and water will follow. That's how it works. So when we're looking at the term concentration, the concentration is defined as the number of solute, which is usually the solid or the amount that's in less, dissolved in the solvent. And the solvent is usually water, or it is the one where there's more. And you end up with a concentration gradient then. Okay. Filtration is a type of passive transport where there is pressure moving the water. So if you filter out, say, your coffee in the morning, the pressure of gravity will draw the water through. Usually in the body, when we're talking about filtration, we're talking about the pressure of the blood. So blood pressure. Blood pressure will be the force due to the pumping of the heart, and it moves blood through the bodies, especially through the kidneys, in a force called filtration. So here's an example. Here's this yellow thing is the kidney, and because of blood pressure, sodium, potassium, and other things will leave the capillary called the glomerulus. Okay. Then there is something called facilitated diffusion. In facilitated diffusion, that is when the substance moves across the membrane, high to low, without any extra energy, but it moves using a carrier channel. And an example is, glucose is the example of facilitated diffusion. So here they have, it's kind of like a rotating door where it moves and you get into the lobby through it. And in the picture down below, you will see the glucose molecule or the glucose carrier, and the glucose will bind. It will carry the molecule inside. Okay? An example of this that I just want you to be aware of is cystic fibrosis, where sodium and chlorine are unable to diffuse. Or another example that's quite a concern to the body is diabetes mellitus. Because insulin isn't there to turn on the glucose facilitated diffusion, glucose cannot be transferred in. Good. That's passive diffusion. Passive diffusion is no extra energy, high concentration to low concentration. The next type we're going to look at is active diffusion. Active diffusion is broken down into pumps. Endocytosis, which is membrane bound, and endo means in, and exocytosis, which is membrane bound, but it moves out. So when you're looking at the pumps are mainly in, the most common one is a sodium potassium ATP pump, and this is used in nerves and muscles to pump out potassium and pump out sodium and pump in potassium, and the energy is moved to set up this concentration gradient by going against the concentration gradient. Okay, that's active. Now another example of moving things in that are too large to go through the membrane is by the process of endocytosis. Endocytosis is when you have the uptake of liquid or food that is too large to go through the membrane, so the substance or the cell wall will form around it, forming a vesicle, and that will move in. If it's a large thing, it will be called phagocytosis. If it's a small, it's pinocytosis. And here is a beautiful example. So in this example, here you have your bacteria. Along comes the white blood cell, which will go around the bacteria and engulf it. Here's an animation of the same thing. And the white blood cell is engulfing the bacteria. Okay, so it's pretty straightforward, but they're large. Then when that's endo, which will pull things in, exo is when you are transporting things out of the cell, and it's done the same way in the fact that a membrane will bind to the cell membrane, a membrane-bound vesicle will bind to the cell membrane and release the information or whatever's there, usually the hormone that's trying to be secreted. So here is exo, we're assuming this is the outside, this is inside. There has been some proteins made inside the Golgi apparatus here, and the proteins will result in it going out. Okay? Oh, we haven't talked about that yet. So now let's look at parts of the cell. So, more examples. When we look at the cell, we've got the nucleus and the cytoplasm. The cytoplasm is the watery substance around the cell, and it contains the organic solids, proteins, carbohydrates, and lipids, inorganic mineral and gases, and it is there to regulate the cellular exchange within materials. Then we have the nucleus, and we talked about the nucleus. The nucleus dictates the energy or the activity of the organelles. It is double-walled. It has pores, which allows things in and out. It contains chromatin, which is another word for DNA or chromosomes. Inside the nucleolus is a special area called, inside the nucleus is a special area called the nucleolus. And the nucleolus is an area where there is special RNA molecules made. Then there's something called a centrosome. Now the centrosomes they kind of look like little stacks of wood, and they're involved in cell division. So you may have heard of mitosis and spindle body fibers, or we'll look at it at the end of this chapter. Here is your centrioles. They will migrate to the other poles, and your spindle bodies will form in the process of mitosis. Good. Next, you have the mitochondria. The mitochondria is the powerhouse of the living cell. And this is, if the cells need more power, then there'll be a large number of mitochondria in it. So an example is a cell that makes enzymes. It will have a large amount of mitochondria in it because it needs a lot of energy. So the mitochondria is the powerhouse. Then attached to the mitochondria are flattened folded sacs. And these flattened folded sacs have ribosomes on them, and they are rough endoplasmic reticulum. And this is where the proteins are, the ribosomes is where the site where proteins are synthesized. Then the proteins will go into the rough endoplasmic reticulum for modification and packaging. Then there's something called the smooth endoplasmic reticulum. In the smooth endoplasmic reticulum, there are no ribosomes, but this is responsible for where lipids and steroids are synthesized. And then once they are done, they will leave the endoplasmic reticulum in vesicles. So when you look at it, here's our mitochondria. They're double membraned, kind of look like peanuts. Here is our nucleus with our nuclear membrane. Nuclear pores, and attached to the nucleus is the endoplasmic reticulum. If there's dots on it, it's the rough endoplasmic reticulum. If there's no dots, it's the smooth endoplasmic reticulum. Then they will pinch off and form these vesicles. These vesicles will then go and create a new organelle called the Golgi apparatus. The Golgi apparatus are also flattened sacs. And this is where it will process and store proteins. And then this will, the Golgi apparatus is sort of seen as the post office. And this is where things get processed and get ready for shipping to leave the cell. Some other organelles we're going to look at is the lysosomes. So lys rhymes with slice. So these are the bodies that slice. So lysosomes contain powerful enzymes which will then fuse with this membrane that's been created by endocytosis, and the lysosome will break down whatever in the vesicle. Lysosomes help us keep healthy. Good. There's other organelles such as the cytoskeleton, and the cytoskeleton is skeleton inside the cell. So this is a network of microtubules or filaments that will allow the cell to maintain shape, but it also allows for mobility and some messaging. Flagella are whip-shaped tails like you see on a sperm. And cilia are short hair-like projections that will wave particles of motion, will move particles in a wave motion. And this is an example of things formed on your throat and also in the oviduct. Now we're going to get into the cellular processes inside the cell, and the first one is cell division. Cell division is the process of making new cells, and there's two types of cell division. One where they just divide, and it's called asexual reproduction, and you make identical copies of themselves. The process of cell division or asexual reproduction is also called mitosis. So mitosis is a very organized process where the organelles are sorted, the DNA or the chromosomes are sorted in a very particular and organized way. And this occurs in all eukaryote cells, and a eukaryote cell is anything with a nucleus. So when you look at the life cycle of a cell, it's called the cell cycle. Most of the time, the cell is sitting in a growth phase, which is called interphase. And it's performing normal functions. It's not dividing. When the cell needs to reproduce, it then enters the mitotic phase. And the mitotic phase has two portions, mitosis and cytokinesis. So inside the mitotic phase, there's 4 different processes. And they're phases and they're continual phases. But there's specific things that occur in each one. Then at the end, once the chromosomes have divided, you get something called cytokinesis, which is the division of the cytoplasm at the end. So when you look at mitosis, it goes prophase, metaphase, anaphase, or telophase, or I remember it as PMAT. So when you look at prophase, prophase is the nucleus is disappearing and the chromosomes become visible, centrioles line up. So let's look at the picture here. Here's prophase. It used to have a nucleus here. The nucleus is gone. The centrioles are at the end. The chromosomes are lining up and these spindle bodies form. Prophase. Next is metaphase. In metaphase, the chromosomes are in the middle. Middle. Then in ana goes away. So in anaphase, the chromosomes, which were paired up, are now being pulled away. Telophase is when they come together to form a new nucleus. And cytokinesis is the division of the cytoplasm. So to go back, prophase, the nucleus disappears, the chromosomes become visible. The centrioles move to each side and spindle bodies form. In metaphase, the chromosomes line up. In anaphase, the chromosomes split apart or move away. Away. In telophase, the chromosomes move to the end of cells, the spindle fibers disappear, and the nucleus reappears. Now, cytokinesis happens right after or kind of during the last bit of telophase, and this is when the cells divide in half, the cytoplasm divides in half, and the organelles divide in half, and you end up with the original cell, which was called the mother cell, now forming 2 identical body cells. So this is normal cell division, and this is mitosis. The purpose of mitosis is to replace, repair, regenerate, and growth. We're going to talk a little bit about cancer and pathogens, but I'm running out of video time, so we will do that in the next video.